And now it's time to do some QC. So it is very important to do the QC because we would like to see the quality of the cells and which type of filtering we need to apply on this data set before going to downstream analysis. There are given metrics that we can generate, for example, total count, which tells us the total number of molecules in a cell, gene by count, which refers to number of genes with at least one count in a cell, in a way showing us the expression level. We also calculate the percentage of mitochondrial genes, ribosomal genes, and hemoglobin genes, referring to blood cells. So again, we create a new column in the var slot, and then we say which of these genes, starting with empty, to be labeled as true in this column in the A data var. So if we do like this, we get this one. The same information up to genomes, but then at the last three columns, now we have true or false for mitochondrial, ribosome, and hemoglobin genes. So it's just labeling whether a given gene is coming from the mitochondrial genome, for example, or they are labeled as ribosomal or hemoglobin. These are the reg expression that you can use. I'm not going to dive into that. But anyway, now that we have these, we can use Scanpy to calculate the metrics. So just an overview of what we discussed in the lecture is that in Scanpy you have different modules. So you read and write, which we talked about that, and then we have three more modules: preprocessing, tools, and plotting. So now we are going to use the preprocessing functions in this stage. So what we do, we call calculate QC matrix. This is our data object, and the QC vars are empty, ribo, hemoglobin. And then we do not do any conversion whatsoever. Now you can see that in data in the var we have the gene ID features corresponding to what we had before, the column for mitochondrial, ribosomal, hemoglobin, and these new metrics. One thing that we can do is to check the content of this. Now here you can see that we have the values for each of these metrics. Now let's do some plotting. We can use violin plots. So violin function, the object and the features that we are interested, some settings in order to see the distribution of the cells across the violin distribution that we have, and then we group by samples, rotation of the labels by 90 degrees, and we don't want to have it as multi-panel, just in one single panel. There we go. So gene by counts, here we have a slight shift, but what we have here is total count corresponding to total UMIs, and then we have the percentage of mitochondria, percentage of ribosomal genes, and hemoglobin. So in the first glance you can see these two samples, COVID 15 and 16, tend to have lower gene by counts. We will come back to that. In addition to that, if the total can look okay-ish, and then in the percentage of mitochondria, we do see some differences already. For example, COVID 15, 16 tend to have cells with higher mitochondrial expression. So at the end of the tail of distribution, we see a larger cloud of the cells. And COVID 17 as well, which shows lower percentage of mitochondria, which is not a bad thing. I will talk about that in next few minutes. Anyway, now the ribosomal look okay-ish. We have mostly this looks okay. If you want to be picky, you can talk about the differences in the distribution. But for now, I think we are good, and we have the low percentage of hemoglobin, but we are going to filter these genes as we move on. So it's nothing to be worried here at this stage. So as I told you, COVID 15 and 16 behave a bit differently. But let's look at this in another angle. So we would like now to plot total counts versus percentage of mitochondria using the plotting module and the scatter. We have the object, the X and Y values, and then we color by sample. So we have total counts in X-axis and the percentage of mitochondria on Y-axis. So the first thing that you can see is that the samples that have high total count tend to show lower mitochondrial percentage. And for some samples, since we are plotting all up on top of each other, we cannot pinpoint exactly which sample, but we can see for control 19 and also COVID-17 tend to be the same pattern. But for the rest, we can see here and there, for example, 15 and 16 tend to have higher mitochondrial genes.